Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. We have discussed two steps of light reaction, that is absorption of light and photoexcitation of chlorophyll molecules, and photolysis of water and evolution of oxygen. We will take up the third step and the most important step, that is mechanism of electron transport. As I have already told you, this electron transport, it involves three thylakoid protein complexes. Three thylakoid protein complexes, that is pigment system second, which we also call a light harvesting complex second, cytochrome BF complex, and number third is PS first, that is also called a light harvesting complex first. Light harvesting complex first. Now we will see how this electron transport takes place. Let us take first pigment system second. The chlorophyll and the other accessory molecules of light harvesting complex second absorb light in the form of quanta and transfer their energy of the molecules of and transfer their energy to the molecules of the special chlorophyll A molecule, that is P680, a special chlorophyll A molecule which acts as the reaction center for light harvesting complex second, that is known as P680. All the accessory pigments and antenna molecules, they absorb sunlight and transfer their energy to this reaction center by resonance transfer. The chlorophyll molecules of P680, after absorbing sunlight, and when sunlight strikes this chlorophyll, special chlorophyll pigment system second, or light harvesting complex second, after absorbing light energy, it dissipates their energy in the form of electrons, to the nearest electron acceptor, that is phaeophyton, which is the first electron acceptor. And when this electron, it moves out of this P680, a hole is created in P680 due to the loss of electron, and this hole is filled by the electron which comes from the photolysis of water through tyrosine residue, manganese protein, which is also called as tyrosine residue. It is essential because until this hole is not filled, the pigment system second, that is P680 molecule, will not absorb further energy, and thus photochemical reaction will stop. It will not take place. It would have come to halt. It will stop. So what happens, this electron, when light strikes this pigment system second, excitation takes place, an electron is expelled out, that is received by the first electron acceptor, that is phaeophyton. A hole is created. This hole is filled up by the electron of water molecules already split via tyrosine residue. From phaeophyton, from here, electron is transferred to another electron acceptor, that is known as quinone or plastoquinone. It is then reduced to plastoquinone. The plastoquinone molecule has very importance in this whole process. It is the molecule which takes 2 electrons and 2 protons for its full reduction, and it needs 2 electrons and 2 protons for its reduction. 2 electrons and 2 protons. In electron transport chain, two quinone molecules, that is quinone A and quinone B, this are present. It takes electrons from phosphatin and protons from stroma, and which are already found in the stroma, these protons, which also come from the photolysis of water. Due to its vectorial arrangement in the membrane, it is half embedded in the membrane and half in the stroma portion. After plastoquinone is fully reduced to plastoquinone, it changes its position in the membrane. Now it is half embedded in the membrane and half in the thylakoid membrane. In this particular position, it gets oxidized back to quinone by dissipating two electrons one by one to cytochrome BF, the cytochrome B6F complex, and two protons in the thylakoid lumen. After oxidation, it returns back. Now from this, we can see from quinone this electron comes to cytochrome BF complex. The cytochrome BF complex contains two cytochrome B, one cytochrome F, and an FES protein. Then the electrons are transported from this cytochrome BF to an electron, a mobile electron carrier, that is known as plastocyanin. From plastocyanin, this electron, it is transported, it is received by the pigment system first, that is P700, because when light strikes this pigment system second, it also gets excited and a hole gets created, and this hole is filled by the electron which comes from this electron transport. That is this pigment system P700, or what we call a pigment system first. This having another this complex, that is known light harvesting complex first, and here the reaction center is P700. What happens to this P7? It gives up electron to the nearest electron acceptor, that is a chlorophyll molecule A-like, and that is the FES protein, an iron sulfur protein. This is the iron sulfur protein. It receives this protein. Now from there, here the electron moves to another electron carrier, that is ferridoxin. Then it reduces NADP to NADP with the help of the enzyme ferridoxin NADP reductase, and one electron from ferridoxin, it moves to cytochrome B6. From cytochrome B6, it moves to plastoquinone. In this way, here cyclic this electron transport takes place, and on the other hand, the electron which moves from PS second, it ultimately reduces ferridoxin to ferridoxin. That means it forms a non-cyclic electron transport chain. In this way, we see that electron transport chain takes place. Electron transport takes place two ways. One is cyclic and another is non-cyclic electron transport. In this whole scheme, we see non-cyclic electron transport as Z, and English alphabet Z-shaped this electron transport chain gets produced. Z-shaped this structure gets produced. That is why we also called it as Z scheme. This is all about the electron transport. Inshallah, in the next lecture we will study photophosphorylation. Thank you. Assalamu alaikum.